lapel.
Welcome to our YouTube video on the fascinating world of bioinformatics. In this video, we will explore the intersection of computer science and molecular biology, and how it has revolutionized our understanding of biological data. So, let's dive in. Bioinformatics is the interface between computer science and molecular biology. It involves the use of information technology for the analysis of biological data sets. At the core of bioinformatics is the database, which supports data input and management. Data input requires the generation of data using experimental techniques such as DNA sequencing. On the other hand, data management relies on hardware and software developments to store and organize a vast amount of biological data. The emergence of genome sequencing required the development of IT systems to handle the output of large-scale sequencing projects. This led to the creation of data warehouses, which store digital data, and data mining, which involves interrogating databases of various types. To make sense of the sequence information, several requirements must be met. Accurate generation and capture of information, clear and logical presentation of the sequence in a visual format, annotation of the sequence for orientation and identification of features, and consistent use of annotation by different users/providers. Nucleic acid databases, such as GenBank, EMBL-Bank, and DDBJ, are repositories for experimentally derived sequence information. These databases, maintained by international collaborations, provide a wealth of primary annotated nucleic acid sequence data. Protein databases, like UniProt, contain entries determined by direct methods or by translation of predicted mRNA sequences from nucleic acid databases. However, it's important to note that derived sequence data may not capture all modifications to the amino acids. Garbage in, garbage out is a phrase in computing that emphasizes the importance of valid and reliable data entry. Bioinformatics tackles this issue through careful data generation, accurate data entry, and rigorous data processing. Quality control measures ensure the accuracy of sequencing data. Computational experimentation, known as in silico experimentation, provides novel insights but does not replace real practical work. For example, translating a DNA sequence into a predicted protein sequence can be done in silico, but experimental identification and analysis are necessary to establish the presence of the protein. Presentation interfaces play a crucial role in enabling users to navigate through the data. Interfaces like Ensemble, Vega, and EndRes MapView provide clear user interfaces with specific functions, such as search tools for restriction enzyme recognition sites and protocols for translating nucleic acid sequence data into proteins. Protein-protein interaction mapping is a powerful technique used by scientists to understand how proteins interact with each other within a cell. It helps us uncover the complex network of interactions that drive cellular processes. One popular method for protein-protein interaction mapping is the two-hybrid assay. In this assay, the DNA binding domain and the activation domain are fused to specific proteins, let's call them X and Y. These hybrid genes are introduced into a cell. After translation, the DNA binding domain protein X fusion protein binds to the regulatory sequence of a reporter gene. However, if protein Y, PRE, does not interact with protein X, BAIT, the reporter gene is not transcribed. To demonstrate a specific protein-protein interaction, the coding sequence for the activation domain is fused to the DNA for protein Z. This construct is then transformed into a cell containing the DNA binding domain DNA X fusion. Now, let's talk about large-scale protein-protein interaction studies. In these studies, two libraries are prepared, each containing thousands of cDNAs generated from total cellular mRNA. Method 1 involves screening the library for the production of proteins that interact with a hybrid BAIT protein. This is done by introducing individual BAIT DNA binding domain constructs to arrayed clones by mating. Method 2 is a bit different. Each yeast clone in a BAIT library is mated en masse with a mixture of strains in the PRE library. Positive interactions are identified by screening for activation of the reporter gene. Now, let's shift gears and talk about molecular databases. These databases, such as GenBank, store and manage sequence data. They have become invaluable resources for researchers in genomics and proteomics. Have you heard of metagenomics? It's the study of genetic material recovered directly from environmental samples. By analyzing the DNA from various microorganisms, we can gain insights into the diversity of microbial communities. Functional genomics focuses on patterns of gene expression. Techniques like DNA microarray technology and serial analysis of gene expression, SAGE, allow us to track gene expression levels and identify genes that are up or down regulated under specific conditions. Finally, let's talk about proteomics. This field helps us understand the proteins present in a cell or tissue and their interactions. Techniques like 2D PAGE, protein microarrays, and protein expression profiling provide valuable information for studying diseases and developing therapies. And there you have it. Protein-protein interaction mapping, molecular databases, metagenomics, functional genomics, and proteomics are all powerful tools that help us unravel the mysteries of the biological world. And that concludes our overview of bioinformatics. We hope you've gained a better understanding of this exciting field that combines computer science and molecular biology. Thank you for watching, and don't forget to like and subscribe for more informative videos. Stay curious.
Pharma Pal.